speaker is Sergey Mangul, who's in the Eskin Lab as well as in the QCB laboratory.
Yeah, that's right. Okay, thanks for coming. So, as we develop the tools in the Eskin Lab, sometimes we get inspired by things which are happening around. This is I saw the other day, and this is called the dumpster diving, which is actually the real term of looking for the garbage in order to find some clothes you can wear or some food you can eat. So I'm not here to convince you that's the lifestyle to follow. Instead, we can borrow some ideas, meaning that we should be always careful when we throw away the data. In this case, it will be so to account for the missing data, we designed the ROP, which is Read Origin Protocol, which I will explain in a couple of seconds. So thanks, Grace, for doing a great introduction for RNA-seq. Actually, that's exactly the same slides we are using. So I will just say that, so given the alignment of the reads, we can solve a bunch of the problems which will be discussed today. But what is in common between all those is that we can only account for the fraction of the reads. And usually the reads which were not mapped onto genome, they will be discarded and then will go to the trash. So we think that's not right. So the project has started as the kind of more exploratory one, just because we were curious to know what among those unmapped reads, and then apparently just kind of going a little bit in front. So there is very many interesting things among the unmapped reads. So we designed the so-called Read Origin Protocol, which looks carefully for the unmapped reads, trying to find some hidden treasures. The paper about the protocol is online. It's called Dumpster Diving in RNA-Sequencing to find the source of every last read. It's on the bioRxiv. And then usually the unmapped reads accounts for 9–15% of all the reads in the current big studies like GTEx or TCGA. So the protocol has two modules. So first, as you have the reads map, which accounts for the majority of the reads in your study, almost around 90%. We have two modules. The first module is gProfile, which classifies the map reads according to genomic features. So given the mapped reads, we can classify in the UTRs, coding regions, introns, and other features. That's useful to benchmark the new sequencing technology as well as estimate the sample degradation. Also, we designed the other module is RProfile, the module which will quantify the reads according to the repeat elements. So this was about the mapped reads. So as we thought about the unmapped reads, the ones which were here in the gray, the first step was the quality control, where we filter out the reads which are of the low quality, low complexity, or the ribosomal reads. And then they account for the 7% of the total number of reads. We also thought about the heuristic implementations of the high throughput mapping algorithms, where those provide basically no guarantee that all the reads were mapped due to the speed. And then we were able to identify, using the most sensitive alignment, which is the megablast, we were able to identify around 5% of the reads which we think are human in origin. We call those lost human reads. So if you look at the relative, at the distance of those in terms of the reference, some of them come within threshold of the high throughput mapping we were using, or the top hat. Some of them contains additional mismatches. If you're curious to know from what particular place in the genome they're coming from, there is no particular place. They're kind of randomly distributed across the genomic features. This is the genomic profile of the poly-A data based on the mapped reads. And then this is pretty similar as we would estimate the genomic profile from the lost human reads. With some exception. For example, we see among the lost human reads, we see increased number of intergenic reads. Consistently, that was for the ribodepletion libraries. Okay, so the first step of the protocol is to identify so-called lost repeat elements, the repeat reads which were missed by the mapping-based tools. We also thought about the things which are not linear in terms of genome and thus will be not captured by the mapping-based protocols. We consider so-called non-collinear RNA, which are the transplicing and the fusions. So if we think about the transplicing events as the splicing which occurs across the genes. So given two genes, we see the splicing from the exons which belongs to different genes. We call this as transplicing. As to the fusions, those genes belongs to the different chromosomes. Also, the circular RNAs are there where the reads are spliced in the kind of abnormal way in the tail to head configuration. So as you can see here, two exons are spliced this way, which kind of suggesting that that particular transcript was circularized. Okay, so having those, so we're able to identify 0.1% coming from those events. We also thought about the immune component, about antibodies, where, again, things are not linear, where reads are coming from the recombinations of the VDJ genes, and in this case will be not captured by the mapping-based protocols. We use the IgBlast to identify those genes, which accounts for 0.02%. We also working on developing a special tool to do that, to increase the speed, because so far this process is pretty intense in the computational time and speed. We look for across multiple tissues for 3,000 RNA-seq samples from the GTEx, and on the y-axis you see the number of actually cut it. So it's the number of VGA recombination of the immunoglobulin kappa locus, where we see increased number in the number of recombinations in the spleen, some high number, surprisingly actually high number in the colon, and some other tissues. This is the project which is in progress, so we're exploring and trying to make sense of the data. And the last step, as we account for all possible reads with the human origin, we thought about the non-host component. As you might think of, every sequencing is a metagenomic sequencing, so we were able to observe 0.2% of the reads coming from the viral, bacterial, and eukaryotic genomes. Often insufficient number where you can reconstruct the taxonomy of the bacteria or detect particular viruses which is in your sample. So given that, we are able to account for 99% of your data, coming with 1% being unaccounted. We don't know the origin for that. So we think those might come from the template jumping, where we actually see among those reads, some of the reads are of the kind where you have half of the read coming from the human and half of the read coming from the worm. So if you think the worm cannot integrate your genome, that's coming for the sequencing artifact. As well as there might be some unknown microbes where we design the method of condensing the k-mers into the de Bruijn graph and then comparing the sequencing structures directly between the samples, which allows you to compare the microbiome in the reference-free way. Okay, so one more thing about the microbiome. So we're exploring the GTEx, where we have 33 body sites, and then we're curious to see the microbiome distribution. So far, we are happy to see nothing in the brain, in the heart. Those, as the contamination is a huge issue, we're planning to use those kind of as a negative controls to control for any contamination coming from the reagents or coming from the spontaneous contamination introduced by technician. Okay, given that, so I hope I was able to convince you that RNA-seq is more than the gene expression and the transcripts. It can, especially the unmapped reads, contains valuable information. So if you would like to profile the immune response, you might look at the VDJ recombination. If you're interested in microbiome component of your data, look for the microbial reads, as well as circular RNAs are believed to be very important in the diseases, especially in the they often occurs a lot in the brain. So we designed the software, which is called ROP, Read Origin Protocol. We have some tools which we develop as well. We take some other state-of-the-art tools. So the tool comes with the no pre-requirements. So you just download the tool, and actually the most complicated part, you just need to download gigantic references where we map the reads onto, and you should be good to go with no installation. And this is kind of the typical output of the ROP, where in every single step you will be provided how many reads are mapped, how many reads are low quality or low complexity, how many reads are lost human reads coming from the B lymphocytes, and so on. And we have a detailed tutorial being available on the wiki, where we have one sample, RNA-seq, where we provide every single step and the expected outputs, so it will be pretty easy to learn how to use the tool. And we're exploring some other technologies besides RNA-seq. We got a request from Australia about the ChIP-seq. People were interested in the profiling repeats from the ChIP-seq, so they want to use the ROP to look for the repeats among the unmapped reads. So again, the tool is available here. And I would like to thank Elazar Eskin and then Noah Zeitland from UCSF and Sagiv Schiffman for leading the project. And then I will be happy to answer the questions. Thank you.